So I will, of course, cover some of the same turf that Marianne did. We think about this problem very similarly, which is why it's always so fun to chat with her about research and these things. So here's my version of the Venn diagram that Marianne showed. I think of brain-computer interface research as sitting at this wonderful watershed between machine learning and systems neuroscience. It's only in the last few years, and mostly because of BCI research, that we've become a data-rich field, which has drawn a lot of interest from people who do machine learning. So here we are. In fact, I think of it more like this, where systems neuroscience and machine learning are already on a continuum. Okay, the ideas that are coming out of machine learning are very much informed by what we know about the brain, and vice versa, with BCI still sitting between these technologies. Credit where it's due, this is the enormous team that I've had the pleasure of working with to do this research. My theoretical / neuroscience colleagues at CMU are here. These are the trainees, postdocs, and graduate students who did this research. Many of them got their PhDs doing this. And actually, Kristen now works with Jen Collinger, whose lab created the Jan video that Miriam showed us. And these well-dressed individuals are the neurosurgeons who put in the multi-electrode arrays. So here's our little map where theorists are mostly over here, experimentalists are here, and we blend together very seamlessly. What I'm proud of about this research is that the theory is embedded in the experiments as opposed to being something that happens with the data after the fact. Okay. I want to ask you a question to get started, and I really do want you to take this question seriously. What changes in your brain when you learn? I would love it if people would be willing to give me some answers to that question. Anyone take the bait? Everyone's thinking about it. What happens in your brain when you learn? If you've picked up a copy, yes? Yep. Yep. You build new synaptic plasticity, new connections between neurons. What else? Any other answers that people have in mind? Many things. The entire brain changes at every level of organization. It's true. Yeah. There is all kind of metabolic processing that goes on. Spiking is fairly cheap, but building new synapses is expensive, involves protein synthesis, and often requires a night's sleep. So the microglia, the same things that destroy our electrode arrays after a time, are busily taking out the metabolic byproducts of plasticity. Yep. Okay. Another answer. So here's the types of answers that people often give. Synaptic plasticity is definitely first on the list. A few years ago, many people, MRI, functional MRI, was really very popular, still is a dominant technique, but very popular a few years ago. So a lot of people had heard about British cab drivers with an expanded hippocampus, or pianists with an enlarged motor cortex. The question in my mind always was, why does that confer greater ability? Okay, it's not a causal explanation, it's an observation. When I asked this question of systems neuroscientists, those of us who study neurons for a living, people usually mention that neurons change their tuning, okay? Individual neurons are selective to events in the visual environment or the somatosensory environment, and or they're active before you make a movement with a high degree of selectivity. So some movements are active for reaches to the right, some are active for reaches to the left, and everything in between. So with learning, these tunings adjust, okay? So these are the types of answers that we usually get when we talk about how the brain changes when you learn. But I want to say that there's a whole new level of plasticity that we can look at thanks to BCI technologies, and that's how populations of neurons reorganize. We don't look directly at networks. We just look at the kind of shadow or the reflection of network activity in terms of how populations of neurons interact with each other. Okay? So it's a relevant level for studying learning because at the end of the day, it's patterns of activity in motor cortex that course down the spinal cord, synapse onto my muscles, and make my arm move in a coordinated way. So it's patterns of neural activity that drive behavior and probably drive cognition. Okay? And it makes sense if we can to study patterns of neural activity in their natural form. And BCI technologies, these 100 electrode arrays allow us to do that, okay? And machine learning is needed to begin to deal with high-dimensional data. The problems that Maryam outlined about how high-dimensional and noisy these data can be, frankly, we need help dealing with that chaos, finding the structure amid the chaos. Okay, 2 types of motor learning that will form the basis for the experiments I want to describe. And I'm hoping I can tap into your everyday knowledge about familiar situations. Here is my Dadmobile. I have to drive it in all kinds of weather conditions in Pittsburgh. And then I come here to Newport Beach, and I can rent the slingshot, okay? So this transition from this thing to this thing, it's the same skills. It's still pedals and a steering wheel. It's an adaptation process. I might be awkward or clumsy at first when I'm driving this thing, but by the end of the day, I don't want to give it up. And going back to this thing requires another period of adaptation. So this is a motor learning experience that's fast, okay? Here's a slow motor learning experience, skill development. You're all familiar with cognitive skills, but of course, many people also develop motor skills. Almost all of you can do this, but if anybody can do this, ride a unicycle while catching things on a, can anyone ride a unicycle? Anyone here who can? Cool. Okay, this is a skill. Even if you can't do it, you can appreciate. She started when she was 7 years old, and basically what you do. If you want to develop this level of mastery with something new, you've really got to stick at it. What is it about the brain that makes adaptation fast, okay, but skill learning slow? So here's a very simple hypothesis, simple to state, but until BCIs came around, hard to test. The hypothesis is this adaptation process reuses existing patterns of neural activity in a new way. This process requires your brain to somehow forge new patterns of neural activity. So the reasons why synapses are changing, the reasons why cortical maps are expanding, the reasons why network connectivity is changing, is to endow you with the ability to express a new pattern of neural activity to give you the ability to perform a new behavior. How do we actually show that? For showing this experimentally, we use Rhesus monkeys. So Maryam showed Jan feeding herself a bar of chocolate. I would bet that there have been a dozen or maybe 24 human beings implanted with BCI devices like the one that Jan has. On the basic research side, the animal research that fuels the development of the human technologies, there have probably been 50 to 75, maybe 100 monkeys implanted with the types of devices that we use in the humans. Okay. The reason why we use monkeys rather than rodents, I mean, the reason why we don't use humans for the basic technology development, I think, is very straightforward. But the reason why we don't use rodents is because the monkeys provide us with a level of behavioral sophistication that's beyond what we can get from a rodent. Okay? So this is just a very brief survey of many of the things that my colleagues and I have used the primate model to understand the neural basis of. Monkeys are able to perform dexterous finger manipulations, eye movement, eye-hand coordination. They have trichromatic color vision. And they have social cognition, as this monkey is saying, don't you dare come near my piece of corn. So all these things that we care about, monkeys do. And my postdoc discovered they also ride bikes. So any questions at this point about where we're going or why we're going there? Enough. It seems like he is, yeah. Yeah. Cheating. He's not really riding a bike. He's riding a tricycle. Stupid video. All right, questions? We're moving on. Okay, so I could watch the monkey ride a bike all day. All right, BCI control. So here is my version of Maryam's slide. We put a 100 electrode array into primary motor cortex. What we care about in particular is the action potential. Who knows, who remembers their college biology? Who knows what an action potential is already? This is the fundamental signaling unit of a neuron, okay? So neurons emit action potentials, which then cause synaptic transmission to take place, and the process propagates. So each of these ticks is a scaled out view of a time when an individual neuron emits an action potential. We have 100 neurons recorded simultaneously, and we use a BCI decoder, which maps neural activity to cursor velocity in 2 dimensions. Okay, for the robot video, it's 10-dimensional control, but for the experiments I'll describe, it's 2D control. Okay, so that's a BCI system. Animal sees the cursor move, and what does he do? He adjusts his neural activity to achieve the goal that he wants. So a BCI is neurofeedback. You're looking at a 2-dimensional projection of your neural activity, using that to drive your neural activity into a more advantageous state for the task you're trying to perform. Looking at your hand and arm move is also neurofeedback. It's just a very complicated effector, and here we have a much simpler effector, which creates the possibility for the animal to learn how to control it. Okay, so I've hinted at a lot of this already, but unlike studying arm movement control, when we're using a BCI, we have all the neurons that directly control the cursor under our observation. When you put an electrode into motor cortex, good luck finding a neuron that actually controls the muscles. Okay. The mapping from neural activity to movement is linear, low-dimensional, and something we've defined ourselves, and we can change it. Okay. The feedback is under our control, and importantly, the animal's arms are at rest while they do these experiments. The only feedback is visual, which means we're able to study cortical neural processing dynamics in their own right without having to worry about contaminating influences from the sensory or motor periphery. Okay, so if we want to use this device to study learning, because learning is intrinsically interesting, what we can do is change the decoder, observe how neural activity patterns change. Lots of people have done this. By lots, I mean 3 or 4 other labs, including Miriam's colleague, Jose Carmina. But what we can do now is change the decoder in ways that trigger fast or slow learning and observe different types of neural activity patterns, reorganizing to accomplish those 2 different types of learning. Okay? Questions? Yeah. Yep. We haven't. A colleague of mine named Nico Hatsopoulos at the University of Chicago has given the animals are controlling a robotic hand that the monkey is grasping. And as the monkey's hand is moving under brain control, he's receiving proprioceptive feedback. And then Nico can break the association by scrambling the hand movement, and that impairs BCI control. So there are labs that are getting at it, but not in the project I'm describing. Yes? Yep, velocity, sorry. Yes, yep, so it's 2 dimensions of speed, each decoded separately from the same neurons. It's just a 100 by 2 matrix, and we pull out the X and Y speed. Hmm? Yes, they're designed to do so. 100 dimensions of neural activity, 100 neurons, independently controlling X and Y speed, and the animal can steer the cursor around with that. And I'll show a video soon. Cool, okay? So, how do we build BCIs that trigger fast and slow learning? I'll describe the recipe here. I'm going to do it geometrically, okay? So in order to be able to represent things on the slide, imagine the animals always did 2D cursor control, but imagine it's just 1-dimensional speed, okay? And imagine we only have 3 neurons controlling the cursor. That's not the real situation, but it builds the intuitions. And everything I'll describe is true in the full space. Okay, so the decoder is better thought of as a mapping, a projection from a 3-dimensional space to a 1-dimensional space. Okay, so we can approach this problem geometrically. We have a neural state space, and the way this works is that at each point in time, we can count the number of action potentials that occur on 3 different neurons and represent that as a point in this joint firing rate space like that, okay? If you slide this window through time, this is a 50 millisecond window over which we're counting spikes across all the electrodes. If you slide that window through time, you can begin to populate the full neural space like this, okay? And your BCI decoder is just a two-dimensional projection within that 90-dimensional space. I'll draw it as a 1D projection here. So Maryam mentioned the fact that BCIs have to be calibrated first. The way that we calibrate the BCI is by having the cursor drift on the screen. The monkey sits there and watches it drift. He probably thinks he's in control of it, and he probably thinks he's doing a really good job, but it's actually just drifting slowly to the target to the right or to the left, up or down on its own. So when the cursor is drifting and the animal is observing it, neural activity fires in a fairly characteristic way with some noise, so that neural activity patterns over here are expressed for cursor movement to the right. And if we run the cursor to the right 6 time steps, then we'll get 6 patterns of activity that are close to each other in neural space. If we run the cursor to the left, you get 6 patterns near each other and in a different region of neural space. Calibrating the BCI is simply drawing a line between them. Okay? It's actually a Kalman filter. It's equations like the ones Maryam showed you, but at the end of the day, you can think of it in these terms. So if the animal wants to move the cursor fast to the right, there's a redundant family of neural activity patterns over here that achieve that goal. If he wants to move slow to the right, he's generating neural activity patterns here. Fast to the left are these neural activity patterns. We evoke learning by rotating that mapping in neural space. Okay? So patterns of activity that used to move the cursor to the right now move it to the left. And if the animal wants to move the cursor to the right, he's got to generate neural activity patterns that used to have no impact or very small impact on cursor movement. So we think of a BCI as a way of requesting new patterns of neural activity and asking whether the animal can learn to exhibit those patterns of neural activity by coupling them directly to a motor output. Okay, so when we do this representation of population activity in a high-dimensional space, we actually find that neural activity explores a low-dimensional manifold within that high-dimensional space. Who is familiar with principal components analysis? PCA, everyone's got PCA. Okay, we use a flavor of PCA called factor analysis, and it is preferable for neural data for reasons that I'll skip, but you can ask me about later. Okay, and this just shows that the low-dimensional projection does not capture the data perfectly, of course. That's the same intuition as you have with PCA. It would require additional dimensions to capture the full structure. And we call this an intrinsic manifold just so we don't have to keep calling it the low-dimensional projection of neural activity. And in our experiments, it turns out to be about 10-dimensional, which is an interesting fact in its own right, because we have 100 neurons that are combining activity to control a 2-dimensional cursor, and they're organizing their own bottleneck, which seems to be about 10-dimensional. Okay? It is what it is, but what we can do is use it to predict fast and slow learning. So Sarah yesterday mentioned the idea of a mental model, and here is our mental model. We have these 3 different spaces, and we can think about how they interact with each other. And there are a lot of creative arrangements of these 3 spaces that can inspire new experiments and a different understanding of the system we're dealing with. So we have the full neural space. We have the low-dimensional space in which neural activity actually resides. And we have the BCI mapping that translates neural activity into neurofeedback, the animal seeing the cursor move. So here is one way to play with those spaces. If we rotate the BCI mapping, forcing it to stay within the intrinsic manifold, this is a situation where the animal's existing repertoire of neural activity is sufficient for good BCI control. If we rotate the BCI mapping so it exits the intrinsic manifold, the animal is forced to generate new patterns of neural activity that he hasn't done already in order to restore good BCI control. Okay, we predict that this is fast to learn, okay, akin to adaptation, and this is slow to learn, more like learning a skill. Okay, videos. So our performance is not as good, I think, as what Maryam just showed. This video is probably 6 years old now. Here's an animal using the BCI that we calibrated at the start of the session that we intended to be as efficient as possible without requiring learning. Okay. Here's what happens right after we introduce one of these perturbations. We can determine the level of impairment that the perturbation introduces. We can make it really hard too hard and the animal just quits. We can make it slightly less difficult, but if it's too easy, the animal has no pressure to learn and we can't really see a signature of learning. So we try to choose something that impairs the directionality and the velocity of the cursor. Here is after about an hour of practice with one of these perturbations. This is a within-manifold perturbation, one of the ones we expected would be learned quickly, and it was. Okay. And then here is an outside-manifold perturbation. After using this thing for about an hour or two, the animal is still impaired. There's hardly been any improvement in performance, which is as we predicted, because the solution lies outside of the existing patterns of neural activity. Okay. It's occasionally frustrating and exhilarating to watch how stuck he is. He can get so close to the cursor. He only has 7 seconds to get there and then just, not manage to close that last little distance for some reason. Great, so a little bit of a quantification. A within-manifold perturbation, when it's introduced, the learning happens fairly rapidly, and there is a washout. So you go back to the old decoder, and there's an after effect. Outside manifold perturbation, learning is impaired and it stays bad. You switch back to the old decoder, and then it picks up where you left off. Mike, go ahead. Yep. Yes, the answer is when you build the perturbation, we do it without regard for the dimensionality, although we are actually thinking of doing an experiment closer to what your intuition is right now. It turns out that all 10 dimensions can show some learning. Okay? We hoped it would be the case that the dimensions that showed the most learning were the ones that became more important for that decoder, but we didn't get that result. So all we know is that all 10 dimensions can show learning. Okay. Other questions? This is a good time for questions. Here is a population quantification. We did this 30 or so times in 2 animals. Yeah? He's not moving a thing. He's just thinking. He's trying to figure out, he's exploring some space of neural activity, trying to figure out what pattern is going to move the cursor. Yeah. Yeah, all we've done so far is to show that the fast learning happens within about a day, So this result is all within a single day. And you can see that some of these outside manifold perturbations do show some learning, but for the most part, they're separated. Slow learning, I'm about to show you, takes more than 1 day, usually 5 or more days. So there's a big gap between the fast and the slow in this study. Okay. Sure. Sorry. Okay. So where we are so far is that some BCI mappings are learnable within a single day, while others take much longer, okay, are not learnable within a single day. I'm about to show you they take longer. And the key predictor is whether your existing repertoire, your existing patterns of neural activity are sufficient to control the new device, the new mapping. Okay. So here's the experiment that we did next. We simply let the animal struggle with the same outside manifold perturbation for a week. And you can form a guess about whether or not that works. I'll show you the data now. Here he is after 1 day. This is a particularly strong perturbation, so his success rate is about 40% correct. He valiantly struggles away at it. Even though we know it's futile, he's not going to get anywhere. He keeps trying. Okay. Comes back the 2nd day, and things are worse. Maybe he's lost some motivation. I'm not really sure. On the 3rd day, performance improves again, and it began to look like there was going to be an uptick in performance. And after that, for the 4th and 5th day, back down. So I'm not saying he wouldn't learn it eventually, but it was too frustrating. Frustrating for the postdoc, Emily Obi, frustrating for the animal, I'm sure. So we went back to the drawing board and decided to look for a way to facilitate BCI learning. Okay, so now you can think about the geometrical spaces I'm talking about. We have the intuitive mapping, which is easy to control. We have the outside manifold perturbation, which is lying somewhere in high-dimensional neural space. And what we did was to divide the angle between them angles, actually, 2 angles, into 5, hopefully easy steps. And on subsequent days, we gradually step the animal through these 5 intermediate steps. So we call this neural coaching. And here is the result of that process. In this video, it takes a couple of minutes to run. I'm only showing one of the targets, the one that showed the largest impairment and the most learning. But this target was selected from among 8 randomly chosen on each trial. Okay. So here the perturbation's been introduced, and as you've already seen, the animal struggles to control this cursor. He can get there eventually, but it's not straight and it's not fast. Okay. 5 days. So in between, we've done coaching. We've done all these intermediate steps, and here we are back at the full decoder. And he's getting there. Clearly feels different from the day 1 performance. And day 14, he's almost back at baseline. So he learned. It took him 2 weeks, and we coached him to help, but he did eventually get there. Okay. Quantification showing that there are some fits and starts, but for the most part, after 5 to 15 days, the animal gets there. He can learn these things, but it's not easy. Okay? And it's consistent with our hypothesis that easy-to-learn mappings can form to the intrinsic manifold. The difficult-to-learn ones require neural activity patterns that are new for this animal. Okay? And then someone mentioned that learning engages the whole brain. It does at every level of organization. Synapses change, but I think this is why synapses change. Okay? We study motor cortex, and we've got a bottleneck through which learning must happen because those are the only neurons that can control the BCI behavior. But there's no doubt that learning engages the entire brain, basal ganglia, thalamus, cerebellum, prefrontal cortex, all engaged, I'm sure, in this motor learning task. Okay. I have more I want to say, but how am I on time? Can I keep going? We'll save questions for the end. I'm going to do a little bit more. So what I've shown you so far is a behavioral manifestation of learning, but because we have all the neural activity, we can look at it offline and try to examine the patterns of neural activity that change when the animal learns. This is a long story, which I'll do very quickly and hopefully do it justice. Okay, so first, how are within-manifold perturbations learned? I already told you that you can do quite well just using your existing repertoire in new ways. And let me just illustrate that point. Okay. So the perturbation compels the animal to learn to use his neural activity patterns in a new way. One possibility is that when the rotation happens in neural space, the animal simply rotates his neural activity to line up with it again. Okay, we call this realignment. Okay? It's as if the latent space that we found using factor analysis represents control knobs, and the animal can just dial in different amounts of those control knobs. The other possibility is that there's less of a change in terms of neural activity patterns. The animal keeps generating neural activity patterns from the same family. He just figures out how to do it appropriately for the new behavior, and that's what happens. So when the animal wants to move right, he used to use these patterns, but now these patterns would move the cursor left, so he stops using them to move right, and he starts using them to move left. Similarly, patterns that used to move the cursor left would now move it to the right, so the animal figures that out pretty quickly and starts using those patterns when he wants to move the cursor to the right. This is what happened, and let me show you that quantification. So what we are doing now is looking at these neural activity patterns, and the way that I can put them on the screen is by projecting them through the BCI mapping. So this is the velocity that would have been exhibited by that neural activity pattern, and it's a way of visualizing a 10-dimensional phenomenon on the screen. So here is the space of neural activity patterns prior to learning. Here it is after learning. The overall distribution hasn't changed. All that's happened is that the animal has figured out when I want to move right, I subselect a different set of patterns than I used to use. When I want to move left, I subselect a different pattern. I throw some away, I pick up some new ones, and we call that neural reassociation. Okay? That's how you learn a within-manifold perturbation. Okay? And let's quickly look at how you learn an outside-manifold perturbation. It's quite different. So here's that same neural space that is the low-dimensional projection onto the cursor velocity space of a 10-dimensional, actually 90-dimensional neural activity pattern. Okay? This is the space of neural activity patterns that are exhibited before learning and with 8 days of learning. Here, for this particular target, is the family of neural activity patterns that are exhibited after learning. These are new neural activity patterns. They lay well outside the distribution that the animal exhibited before learning. Okay. Not all targets are learned that way. This target, now the teal one in this little subplot, this target was learned using a reassociation scheme, and that didn't change over 8 days. Okay, so there we are. So low-dimensional structure is present in populations of neural activity. Lots of people have looked at this lately. Okay? We're showing that that structure actually predicts learning rates, with fast learning simply being a reassociation from the existing repertoire, and slower learning requiring forging new patterns of neural activity, which seems something the brain is reluctant to do, but can do given enough time. And all of this was made possible. I think that that's how motor learning happens for adaptation and skill learning, but it's much harder to show when you have all the complexity of an arm moving in the real world. End of story. Cool. Thank you, guys. And Eddie's going to come take over.